Good morning everyone. I am Dr. Kalpana Sharma, Associate Professor, Department of Zoology, S.P.C Government College, Ajmer. बच्चों, मेरे कल के लेक्चर में मैंने आपको बायोलॉजिकल कंट्रोल पार्ट 1 डिस्कस किया, जिसमें हमने देखा parasitoids and predators which are used as biological agents. आज के चैप्टर में मैं आप लोगों को बताने जा रही हूँ बायोलॉजिकल कंट्रोल पार्ट 2, जिसमें हम देखेंगे पैरासिटिक निमेटोड्स विच आर यूज्ड एज बायोलॉजिकल एजेंट्स. इन पैरासिटिक निमेटोड्स वी आर गोइंग टू सी nematodes, parasites of insect, have also been known since 17th century, but it was only in 1930s that serious consideration was given to using a nematode to control an insect. In 1929, a nematode, Neoaplactina glaceri, was observed infecting grubs of the Japanese beetle Popillia japonica. Parasitic nematodes, which can be studied under classification and biology, pathogenicity and life cycle, dispersal of juveniles, survival of juveniles, natural enemies, insects controlled, advantage of entomopathogenic nematodes, rearing, commercial products, and future roles. In this slide, we are going to discuss the classification and biology. The species of Rhaptida, which is nematoda, Steinernema faltidi and Steinernema scaptersky, and Heterorhabdidae bacteriophora are most important species of entomopathogenic nematodes. All members of order Rhaptida are bacteriophagous, and many of them have phoretic association with insect. The bacterium carried by Starner nematidae is also usually Xenorhabdus nematophilus, and carried by Heterorhabdidae is a species of Photorhabdidus. Pathogenicity and life cycle. The infective juvenile enters the insect host through the mouth, anus, spiracles, or by direct penetration through the cuticle. If the mode of entry is by mouth or anus, the nematode penetrates the gut wall to reach the hemocoel, and if by spiracles, it penetrates the tracheal wall. When the infective juvenile reaches the hemocoel of a host, it releases the bacteria which multiply rapidly in the hemolymph. Usually, the insect dies within 24 to 72 hours. Even though the bacterium is primarily responsible for the mortality of most insect hosts, the nematode also produces a toxin that is lethal to the insect. The infective juvenile becomes a feeding third stage juvenile, feed on the bacteria and their metabolic byproduct, and molts to the fourth stage, and then to male and female of the first generation. Dispersal of juveniles. The juvenile of Steiner nematidae and Heterorhabdidae disperse vertically and horizontally, both actively and passively. Passively, they may be dispersed by rain, wind, soil, human, or insects. Active dispersal may be measured in centimeters, while passive dispersal in kilometers. The fourth one is the survival of juveniles. The infective juveniles do not feed but can live for weeks on stored reserve as active juveniles, and for months by entering a near hydrobiotic state. Natural enemies. Population of entomopathogenic nematodes in the soil are reduced by bacteria, fungi, mites, predatory nematodes, and other soil organisms. Survival is better in sterilized soil than in non-sterilized soil. Mites appear to be especially voracious nematodes feeders. Another one is the insect controlled. Some of the insect controlled are armyworms, carpenter worms, cat fleas, crown borers, root weevils, cutworms, filth fly, flea beetles, German cockroaches, leaf miners, moles, cricket, phorid flies, plum moths, stem borers, webworms, and white grubs. The another one is the advantages of entomopathogenic nematodes. Entomopathogenic nematodes have certain advantages over chemicals as control agents. Nematodes are non-polluting and thus environmentally safe and acceptable, although some countries do not allow the release of non-indigenous species. Another one is the rearing. Steiner nematidae and Heterorhabdidae can be reared in vivo or in nature in insect host, or they can be mass produced in vitro on solid medium. For solid medium culture, a substrate beef or pork kidney or liver or chicken waste may be used. Commercial products. Most of the nematodes based products currently available are formulations of various strains of Sternema carapapaceae and such as ORTHO, Ortho BioSafe, BioVectors, and Exhibit in the USA, Sanoplant in Switzerland, Boden-Nitzling in Germany, and Helix in Canada. Another one is the future role. The futures of nematode-based products for insect control is excellent. The technology used currently for producing, formulating, packaging, storing and shipping nematodes products was developed during past 15 years. Thank you.